In this experiment, we see a rat with an electrode that was surgically implanted into the medial entorhinal cortex of its brain. The rat has been trained to collect food items, which the researcher throws into the enclosure. As the rat moves to collect the food, the electrode records when a grid cell fires, represented here by the white dots. Grid cells are neurons located in the entorhinal cortex. Their firing pattern forms an imaginary hexagonal grid superimposed on the arena floor. As the trajectories of the rat accumulate, the grid pattern becomes obvious. The data from simultaneous recordings of 8 grid cells is used to predict the probability distribution of a rat's trajectory, represented here by the blue dots. The green dots are samples from a null model, or a simulation of the trajectory without taking neuronal activity into consideration. The red cross represents the rat's actual movement. Note that the blue dots accurately predict the location of the rat, while the green dots are randomly distributed. This Nobel Prize-winning experiment shows that a rat's movements can be predicted from the firing of its grid cells. Almost like a map, the grid cells create a spatial representation of the rat's surroundings in its brain, allowing the rat to understand its environment and navigate it effectively.